All right, welcome to the webinar on data analysis for the visual world paradigm using Data Viewer. So some of you might have seen that there was a previous webinar on how to program visual world experiment in Experiment Builder. And so this webinar is going to build on that, and we're going to talk about how to analyze data from that kind of simple visual world template. And these kinds of principles that we'll talk about in the webinar would apply to basically any visual world paradigm. So hopefully this will help in your visual world studies. So in terms of an agenda for today, first I'll just give like a brief overview of Data Viewer, mostly to point you to some more useful information about how to learn the basics of it. Then we'll briefly talk about what the visual world paradigm is. We talked about that in the programming webinar too, but we'll just do a little review. Then we're going to jump into some hands-on usage of Data Viewer. I want to point out that this PowerPoint covers all these different things that we're going to talk about in a hands-on manner. And I thought it would be useful to create a PowerPoint that you can use for a quick reference in case you don't want to watch this video again or try to search for the place in the video. You can like quickly just check out the PowerPoint and go through some of the slides. Let me just give you a little preview so you can see it. It gets into detail about how to do all these different things that we're going to talk about. Sorry, let me back up. So I'm not going to use that PowerPoint to go through Data Viewer because I think it's a lot easier to learn if we just do it in a hands-on manner. But I just want to point out that this PowerPoint will be posted at the same place as the webinar video. So you can check this out and use it as a quick reference. Anyways, in a hands-on manner, we're going to first talk about how to import the data into a Data Viewer viewing session. And we'll just quickly go over some of the basics like of the viewing session once we have a little bit of data imported. Then we'll talk about how to set an interest period in Data Viewer. And this is super important for any eye tracking study. So basically this allows you to focus your data analysis on a certain period of the trial. And in this case, we're going to focus the analysis on a period of the trial like kind of corresponding to the time when the critical word was spoken. Then we're going to talk about some data visualization options. So how can you kind of see visual representations of your data? And then kind of, to me, in a lot of ways, the most important function of Data Viewer is this last thing, which is outputting reports to summarize the data. So this is really how you get like that critical data out that you're going to need for your statistical analyses. And the idea is that Data Viewer is kind of an intermediate step between data collection and statistical analyses. And basically it allows you to do like interest area, like gaze data to interest area matching, and then output your data in kind of a organized format that's going to be friendly with any stats program. So the idea is like you'll import your data into Data Viewer, like do all the analyses you need, visualizations and kind of interest area work and that kind of stuff that you need to do. Then you will output tab delimited text files that we call reports, and then you would import that into a stats program. And we're going to focus our discussion today on two of those reports, like an interest area report and a time course binning analysis report, which actually was specifically designed for visual world experiments. Okay. So let's let me exit out of this for a second because I want to give you a brief overview of Data Viewer. This is Data Viewer, and I have some example data loaded up already. We're going to start from scratch in a second, but basically in this interface we have the inspector over here on the left, and the inspector allows you to navigate through to different trials in your viewing session. So a viewing session is a Data Viewer file. And then on the right, you have the trial view window, and this gives you different visualization options. Okay, if you want to, so that's kind of the basics of the interface here. There are a lot of other things we're going to talk about in a minute, but if you want to get a more detailed overview of how Data Viewer works, then you can log into our support forums here at www.sr-support.com. you can make an account. It's free to make an account, but once you have an account, you can go to this video tutorials section under learning resources. There's also a getting started with data viewer you might want to check out, but if you go to this video tutorials section, you can go to data viewer video tutorial series, and this is a 10-part series. It's about maybe an hour or so long, and you can check this out and get like all the details of the kind of functionality of different aspects of the data viewer software. So you can check that out. And if you're just installing data viewer for the first time, let me point out, you probably should go to this getting started with data analysis, getting started with data viewer. That tells you how to install it and how to, you know, learn about it and that kind of stuff. But it has more detailed instructions for how to get going. Okay, so let's jump into it here. Actually, sorry, let's go back to the PowerPoint really quickly, and let's go through I wanted to go over kind of overview of the visual world paradigm first. So if we start from this slide, so this is an overview of what the visual world paradigm is. Basically, on each trial, a participant will see 2 or more stimuli on the screen. Oftentimes that's 4 images on the screen. Sometimes it can be text, or sometimes it can be videos, but it's often images. Then the participant typically will hear some instructions about how to interact with those stimuli. For example, click on the grapes. That's the example that we built in the visual world programming webinar. And so in kind of our little example template, we have the 4 images on the screen. You'd have one of them would be the target, one of them would be a phonetic competitor, one's a semantic competitor, and one would be an unrelated distractor. And the idea is you're going to like use the kind of look patterns on the different images that are on the screen, like the gaze patterns on those different images, to tell you something about the time course of the processing of the audio that the participant's hearing. And to help us in our data analysis, we want to mark the time of those of the critical audio events of the audio file in the data file. So in this example, we would want to mark like the time when the word grapes is spoken, and that way when we do our analysis, we can kind of lock, we can set our interest period, in other words, to focus in on that particular part of the trial following the onset of the critical word. And then the participant's going to somehow choose which word might be the target, and you might give some feedback. And in our example, we just had like an automatic kind of drift check that would happen at the beginning of each trial. We don't really need to get into the details of that. Okay, so I'm just going to show you an example trial. So in the paradigm that we built in the visual world programming webinar, each trial starts with a cross. When the participant looks at that cross for 500 milliseconds, the images
on the grapes.
The images appear, and you hear that audio. I don't know if you could hear that, but it said click on the grapes. And then the participant would have a mouse, and they would move around, you know, click on the grapes, and then the trial would give some feedback, correct or incorrect. I left that off here. But after we collect the data, as I mentioned, we want to look at the time course of these looks to the target compared to the different competitors and the distractor. And those will give us some indication about the time course of processing different aspects of spoken language. So when does phonological processing happen? When does semantic processing happen? That's kind of like the most basic version of the visual world. People have done some really clever experiments to kind of manipulate conditions, but this is like the most simple version of this task. Okay, so often the goal in data analysis is to end up with a graph that looks something like this, and this is also probably the most basic version of this type of graph. It would typically be binned into kind of segments of time down here that we would call bins. So it would be kind of divided into segments of time, and you would maybe do statistical analyses on certain bins within like the time course of your trial. But anyways, on this graph, you have time on the x-axis, and at some point you have the critical word onset, and you might look at some baseline period for a few hundred milliseconds before the critical word onset, and then after the critical word is spoken, you would be comparing like over time what are the percentage of looks, which is on the y-axis here, to the target versus the other types of images on the screen. And so This is example data. I just totally made this up, so please don't try to like use this for any kind of reference in a paper or anything. But basically, you might expect for there to be a brief bump to the phonological competitor or the grill in our little example as they hear the very beginning of the critical word. And then once they realize, okay, it's not grill, it's grapes, then you'll see more looks to the target. And then this is kind of amazing, but people do actually find this kind of thing, that there will be some kind of later looks even to a semantic competitor compared to the phonological and distractor. So in this case, apple, which would be the semantic competitor to grapes because they're both fruits. So you might expect something like this. And the idea is you can use a time course plot like this to help you kind of look at the time course of spoken language comprehension. Okay, this is just kind of details about the example project that we built. Basically, it had 5 blocks, 1 block of practice trials, followed by 4 blocks of 4 experimental trials. There were 2 counterbalance versions, and I already described what would happen in a trial. Basically, they see a cross, look at that cross, and the images appear. And each trial has those 4 images that we mentioned. And 500 milliseconds after the onset of the images, the audio playback would start. And while the audio is playing back, behind the scenes, Experiment Builder would send a message to the data file at the time of the critical word onset. And that happened because we set up the experiment to do that behind the scenes. And we had, you'll have to like figure out the milliseconds into the audio file for each audio file, the millisecond time point at which the critical word is spoken for each audio file. And you have to figure that out and put it into the data source of the project. But basically that will allow you to send a message to mark the time when the critical word is spoken. And we're going to use that to set an interest period today. And then in this case, the participant got a little bit of feedback. Okay, so that's the overview, very brief overview of what the visual world paradigm is. So let's jump into some of the data. And again, you can use this to get a reference for the different steps involved, but let's not talk through it, let's do it, okay? So we're gonna start from scratch. I have a viewing session opened up already, but let's start from scratch. So first you wanna launch Data Viewer. So, you know, open up Data Viewer. I've already got it open here. And so I'm gonna do File, New, to create a new viewing session. I'm not gonna say, I've already saved this one that I had open. And so let me just point out in Data Viewer, the files that you're working with are called viewing sessions. And so if you do File New, File Open, File Save, or Save As, you're gonna save a viewing session. And typically a viewing session corresponds to an experiment. So we might have a viewing session for a visual world experiment, and that viewing session might have, like in our case today, 4 participants. And so we're going to import all those participants' data into our viewing session. So what we don't want to do is do File Open, because that will ask us to open a viewing session that had been created previously. What we want to do instead is import our data into the viewing session. Okay, so I'm going to go to File, Import Data, Multiple iLink Data Files. And what we're going to do here is point to our, I'm just going to find the folder real fast. So I have a webinars folder, and then this one here, what Visual World and Data Viewer. Okay, I have a deployed folder here, and there's my deployed simple Visual World project. So this is a deployed version of the experiment that we talked about in the programming webinar. So if I point to that folder there and click open, it will list all of the EDF files that it finds in that folder. So we'll search all the subfolders and find the EDF files. And we had this is actually just some example data I collected on myself as the participant earlier. So these are actually just four versions of me, we'll say. But anyway, it's basically, we want to import maybe all of them, but you can choose to import certain ones of them if you want using this dialog. But in this case, I'll just import all of them. And when I do, you'll see a little dialog that tells you the data are being imported, and you'll see the data files appear in the inspector. And so I mentioned that this inspector on the left is what you can use to navigate through the different trials and like participants of your viewing session. Okay, so you'll see all the trials represented here. For a given participant, you can click the plus sign to expand and see all the trials for that participant. And we had 18 trials, so you can click on a different trial to see the data for that trial. Okay, and when you have a certain aspect of the data selected, then you'll see it in this middle panel and you can see the details about it. So I have a fixation selected here and you can see the details about that. Again, I would recommend to see that video tutorial series on Data Viewer for kind of more detailed overview. You can control the which aspects of the data are visible using these buttons up here. So that's the sample trace. These are fixations. This is saccade. So we'll come back to that stuff in a minute. This view on the right is called the trial view window. And this is the window that allows you to visualize the data from your experiment. And whatever trial you have selected will be shown in the trial view window. And it has three main views. First, temporal graph view. Then you have spatial overlay view and trial animation playback view. Okay, so in the temporal graph, let's go to temporal graph view first. In the temporal graph view, you have the time of the trial along the x-axis here, and you have position in pixels on the screen on the y-axis here. And these same buttons that I mentioned control the different control the visibilities of different aspects of the data. So if I turn this on, that's the fixation data. So that means that there was a fixation starting here and ending here. And if I look at the saccades, you can see there's a saccade and then a fixation in this position. By default, it shows the fixation at the x coordinate of the fixation, but you can change that in the preferences if you want. Usually in the temporal graph view, people use the sample data. They visualize the sample data in this view. So if you turn on sample data, notice these buttons appeared on here. If you turn on sample data, you can turn on and off different aspects of that sample data. So this is the x coordinate of that. The red line is the x coordinate of the eye over time on the screen in pixels. And remember, 0, 0 is the top left corner of the screen. So if they were looking up here, this would be 0, 0. And in this case, this experiment was programmed in a 1920 by 1080 resolution, which you can check by clicking on an EDF file. And so if they were looking at the bottom right, that would be 1920 1080 or 1919 1079 actually. But basically, this red line is the x coordinate of the eye over time. The green line is the y coordinate of the eye over time. And you can also look at the pupil size over time using this, velocity and acceleration with these buttons. But you can also turn on and off the interest area visibility, which tells you during this period of the trial, the participant was looking at the semantic competitor. Here they were looking at phonological competitor, et cetera. So it gives you kind of some nice little visualizations of how the trial proceeded over time. And you can zoom in and zoom out using these these little magnifiers here. So that's kind of the temporal graph view. You can save this as to disk using this save trial view as image. Okay, this is the spatial overlay view. And right now we're not seeing the images because we have not set the interest period yet, but we'll come back to it. Basically, this shows us all the data of the currently selected interest period, which right now we're seeing all the data from the beginning of the trial to the end of the trial. And this, in our example project, if you remember from the other webinar, each trial ended with a blank screen. And so we're seeing the last thing that the participant saw in the currently selected interest period, which is the full trial. And so we're seeing the blank screen. And these same buttons control visibility of different aspects of the data. So this is the sample trace. These are fixations. These are saccades. You can see blinks. Maybe I didn't blink. There's a blink. Okay. I probably didn't blink a ton because the trials weren't very long. There's one. Okay, but anyways, you have fixations, saccades, sample trace, blinks, that kind of stuff. And you can also turn on and off the interest area visibility. So each of these squares is is an interest area. And if you want to see detailed information about the interest areas of the trial, you can expand the trial here and you'll see custom interest area set. So these were created automatically by our experiment builder project because we had programmed them into the experiment builder project. I'm just going to go back to it. So we had these interest areas here that moved around as the target and the different types of other images moved around. And so you'll see there, in this case, the semantic competitor was at the top left. Let's select target. Target was at the bottom left, etc. So you can see the different interest areas that way. Okay, and in this, let's choose a different one. So in this case, phonological competitor was at the top left and target was at the bottom right. So basically, I'm trying to illustrate that the interest areas moved around as the image types moved around because we programmed things that way. Okay, so this is spatial overlay view. And this one here, this third button is, so these three buttons control the which visualization option is currently selected. So this is called the trial animation playback view. And in this view, you can see a little gaze cursor here, and you can play back that gaze position over time through the course of the trial. So look at the cross, look at the different images. Okay, settle on the grapes and then get some feedback. So you can select different trials and see the playback for whatever trial selected. You can slow it down if you want. That can sometimes be helpful. So, okay, go in super slow motion here. Maybe that's a little too slow, but okay. We'll see something like that. So you see the eye move around to different positions. You can use this button here to save this playback as a video file. That can be useful. You can put that into a PowerPoint, something like that, to help you with a talk. Okay, so those are some of the visualization options. That's kind of the overview of it. But just seeing this blank screen in the spatial overlay view should be an immediate reminder that we need to restrict our analysis based on a certain period of time, because as you notice in the playback view, we saw a cross, we saw images on set, the subject would hear something, then they would click and they'd see some feedback for a while. But in our analysis, we probably don't really care too much about the gaze position when the cross was on the screen. We probably don't care too much about the gaze position when the feedback was on the screen. What we care about is where they were looking for some brief baseline period before the critical word was spoken, and then up to some period, maybe up until the time when they click on the mouse, or until some amount of time passes after the critical word is spoken. And so that is done by setting what is called an interest period. So this is super important. Whenever you're using Data Viewer, 100% of the time, you always want to be setting an interest period. So you can do this by clicking on this drop down, clicking Edit, and then that brings up the interest period manager. And when you set an interest period, you're going to use typically like the messages that were sent to the data file to start at some time point and end at some time point. And when you set the interest period, it's going to apply to all trials, all participants that are in the viewing session. Okay, so the idea is we've sent messages on every trial that mark these time points, and we're going to use those to set our interest period. So from the interest period manager, you can click this new button. Okay, and the first thing you want to do is give it some meaningful label. So let's set an interest period in our little example that starts maybe 500 milliseconds before the critical word. I'll say 500 pre-crit to mouse click. Okay, that's an example. Okay, so we'll create an example interest period that will start 500 milliseconds before the critical word and it will end at the time when they click the mouse. And in this interface, the top box is where you specify the start event definition. And that can start either from a message from the experiment builder project to the EDF file, or if the host PC were receiving sync signals from another device, you could start it that way. But almost always you're going to start it with a message. And in our case, let's just go back to our little visual world paradigm and experiment builder. We had this message timer crit word that was sent at the time of the critical word. So in our case, it came from this data source column. I've actually changed the value slightly from that webinar, but because this is the actual time when the grapes is spoken. And just a reminder, we weren't in a real visual world paradigm. We would not use the same stimuli over and over again. We would change the trial images and the trial audio file, but and just for example's sake, I just use the same one over and over again. But basically, this timer trigger sent a message to the data file saying timer crit word. And so in our interest period definition, we can type that in just as it was sent to the data file. So I'm literally like typing the same text that's here. You could also copy and paste that. But in our case, we want to maybe have a baseline period that, so we maybe don't want to start right at the time that grapes was spoken, maybe have a baseline period of like 500 milliseconds before that word was spoken. So I'll do a little start event offset of -500, which will push our starting point back earlier in the trial by 500 milliseconds. Okay, now down at the bottom in this box, this is where you specify the end event definition. So this is how we're going to end our interest period. And if we go back to our experiment builder project, you can see mouse response here. Okay, I'll show you in a second. You can actually see these messages in Data Viewer too. I'll show you in just one second. But since I had this experiment builder project open already, we can use that. So when this is the mouse trigger that basically would fire when the participant clicked on one of the images, and it would send this message to the data file. And so we're going to put here mouse response, something like that. Okay. In this case, we could do an end event offset as well, but in this case, I'm just going to leave it at 0. And if I say OK, and then say OK, now we are seeing the stimuli that the participant saw for that period in the trial. Okay, so let me turn on. So that tells you, and if we compare, like if we go back to our full trial period, you will see more data. So we have these extra fixations here, and those go away when we set our interest period. So now we're only seeing data from the period starting at our interest period definition start and ending at our interest period definition end. And this applies not only to what you see in the data viewer interface, but also later we're going to get to how to export these reports from data viewer. It also applies to that. So it's going to restrict any data that is exported to, so that it only includes, so that the reports only include the data from their currently selected interest period. And when you create an interest period, it becomes one of the selections in this interest period selection dialog. And if we want to go back to full trial period, you can just click back to that, and now you're back at your full trial period interest period. Okay, so that's one way we can set an interest period. I'm going to set one more interest period just as an example. So let's go back here, and we have our interest period that we just set here. If we wanted to edit it, we could click this button and we'd see the definition there. I'm not going to change it. I'm just going to say okay. But if we want to make a new one, we can do like this. So we can say 500, what did I call the other one? 500 pre-crit. I'll do the same start to, let's say, 1,000 post-crit. Okay. So what we can do here is timer crit word, and we want to start 500 milliseconds before. So I'll do like that. And then instead of ending our interest period with another message like we did in our first case, notice you can also end the interest period either with another, if you had a sync signal going to the host PC from some third, like maybe, I don't know, MRI or some other device. You probably wouldn't even do it with the MRI, but if you had it going from some other device to the host PC, you could do that. Or if you were using a button box connected to the host PC, you could end based on a button press that's detected by the host PC. If you're using a button box that goes into the display PC, this won't work. But in our case, we're going to end it after a certain amount of time. So I'm going to say duration. And I set up here, we're going to do 500 milliseconds pre-crit to 1,000 milliseconds post-crit. And so that means our total interest period duration is going to be 1,500 milliseconds. And so for our duration, I'm going to do 1,500. So I'm not going to put 1,000 there because that would mean we'd go from 500 pre-crit to 500 post-crit. So this is the overall duration, right? And so if we start 500 milliseconds before the critical word, we need to go 1500 milliseconds to go till 1000 milliseconds after the critical word. Okay, let me point out for some visual world paradigms and some older versions of Experiment Builder, you actually had to use like multiple mouse triggers, like one for each of the types of interest areas. Now there's some new features that don't allow that, as we discussed in the programming webinar. But if you did that and you had a slightly different message, maybe you had a mouse target, mouse distractor, mouse semantic competitor, something like that, you had a different message for each of these mouse triggers, then let me just point out, I forgot to point out this in the message one, but basically, if you wanted to end on any of those messages, then you can make sure that match substrings is turned on, and it would match anything that like you could just do like this, if it had like mouse underscore and then whatever, and then it would match any of your mouse triggers. I just forgot to mention that previously. So anyways, just want to point that out, that in case you had different messages for different possible trial endings, then you can use the match substring option. You can do that for the start event as well. And that way you can match multiple possible like triggers or actions. Okay, so, but in this case, we're setting a duration-based interest period. So we're gonna use this 1500, and we'll say, okay, and now we have a new interest period here. And we said, okay, now we have our data for that interest period. And again, we can change to our different interest periods using this. Okay, so that's how you set the interest period. That's super important. I want to also quickly talk about how you can potentially like adjust interest areas if you wanted to. So I actually kind of intentionally set the interest areas to be like just around the edge of the image. So I made them be exactly the size of the image. If you remember from the way we programmed it in the visual world paradigm, the width and height properties of the interest areas actually get their values from the size of the actual image resource. And so you can see they kind of hug the edge of the images tightly. But let's imagine we wanted to have maybe a little bit of a margin around the interest area. So I wanted to quickly show you how you could adjust the interest areas. So notice up here, I'm going to talk about how we can adjust those. Notice up here first is we have the data. We will notice here that the data are grouped by the EDF file name, by the participant file name. Okay. That's not super useful for us in terms of adjusting the interest areas, because each participant saw the trials in a different order. It was randomized trial order. And what we really want to do is like adjust the interest areas for one trial type and have it apply to all the trials, all similar trials across participants in the viewing session. And so if you remember from the programming, we had a data source column called identifier, which was a unique identifier for each trial type. And so the participants got the trials in a randomized order, but they got the same kind of trials basically. And so what we can do here is go edit trial grouping. And what this would allow us to do is change the way the data are grouped in the inspector. And so we can choose to regroup by any of our data source columns or our variable node values. In this case, we don't really want to regroup by our variable nodes. We probably want to regroup by one of our data source columns, which is identifier, as I mentioned. So basically we can select that, move it over to the right to the selected variables, and then when we click regroup, you can see now the data are grouped by the value of that data source column. And you want to make sure you have a unique identifier for each trial type. But basically, let's expand number one. So we'll see we have 4 trials and see how this exact same trial, grapes up here, grill here, umbrella down here, etc. Okay, so what we can do, first I'm just going to turn off all the I data. So I'm only looking at the interest areas, which the I data we don't want it to distract us, and we also don't want to inadvertently select it. So what we can do is make the adjustment for one of these trials for each group and then apply it to the entire group. So the way we can do that is select these interest areas like this. There are lots of ways you can adjust their size. You can see the other data viewer video tutorial series for more details on this. Like you can just adjust them, select it and adjust them down here, but we can adjust all of them at the same time by selecting them, right clicking, and going to IA shape actions, and we can resize the edges maybe by 50 pixels on each side, for example. That will give us a little bit of margin around the interest areas. Okay, so now they're all a little bit bigger, but notice it still hasn't applied to the other trials in the group. No problem. We've resized them for this first trial. So what we'll do here is click on save the interest area set to disk, and I'll just save it on my desktop. Well, no, I'll give you best practice. So I'll save it in my project folder. So maybe I'll go to webinars, webinar and DB, and I'll just save it in our deployed folder. And you can put them in my files if you want. That's a little folder that's there for exactly the purpose of putting your files in there if you like. But I'm going to call this like 1, and it will give it an extension .ias when I save it. And I'm calling it 1 because it's for identifier 1. Okay. And it says, do you want to add this as an interest area template? And yes, I do. Okay, so if we scroll down, now we'll see that that interest area template is now in our viewing session. And if I click on the overall group of trials here, I can select that template to be the default interest area set for the entire group. And now every single one of the trials in the group has that same interest area set. And you could just, it would take about 5 minutes. You could just do that for each of your possible distinct trials. And that way you can resize them if you want. Okay, but I'm not going to do that for all the other trials. You can, I've showed you the process here. You can do that if you want. But that IA shape actions is just super useful for doing this kind of thing. So that, again, that is, let me select this one. If you go, oops. Oh, I think it's because I have it set to the template, which is, it's already set to template. But basically, we select another trial. Like you can go IA shape actions, and there are all these different kind of things you can do to quickly edit them. Okay, so let's talk one more. Let's do one more quick discussion about another visualization. So I kind of pointed out the main visualizations, but another one you might consider doing is like viewing all similar trial data at once. So at this point, we've been viewing like trial by trial, just one trial at a time. So we're seeing the playback of one trial here. Okay, still in slow motion. Okay, let's view another one. Okay, something like that. So, but let's imagine we want to view all the data from this entire group of trials, similar trials across subjects. Then we can select this trial group and we can turn on what's called the aggregate mode here. And it gives you just a little warning that you're going to see multiple trials data at the same time. And if you do that, then when you play it back, notice we see 4 gaze cursors. So sometimes we call this the swarm of bees. So we see those moving around. That way you can see kind of like all the data. We can select a different trial group. And it can be kind of nice to make these little playbacks as well. And if you want, you can turn it into a dynamic heat map, which is kind of cool to visualize how kind of gaze durations on the different objects change over time when you kind of combine across the trials in the group. So that's kind of cool. You can also do static heat maps by just selecting one of the trials in the group in the spatial overlay view, right click and do create fixation map, or use this button, and that'll create a static heat map. But anyways, I'm going to turn that off for now because I just wanted to show you how that works. So that's how that's basically how you can adjust interest areas, visualize the data, that kind of thing. To me, the most important function of Data Viewer is its ability to output the reports. Okay, so that's done from this analysis reports. And basically, this is what allows you to output tab delimited text files that summarize the data, and that, as I mentioned, can be imported into a stats program for statistical purposes. So Data Viewer is not going to do really the like inferential statistics. It's not going to do any inferential statistics for you really. What it's going to do instead is prepare files that then can be imported into MATLAB, R, SPSS, Excel, whatever stats program you're using to do the statistics on the data. And it's great for this because it has a lot of different output report options. Okay, so if you go analysis reports, you can see all these different report options. And for these up here, the name of the report tells you what each row in the output file will be. So if you create a fixation report, then each row in the output file will be a different fixation in the experiment. And it will all these reports are going to include all trials, all participants by default. And if you create an interest area report, then each interest area for each trial will be a separate row in the output file. So if you create a trial report, you would have one row per trial in the output report. So if we have 18 trials times 4 participants, we have 72 rows in the output file. Okay, for example. So you have all these different report options, and then down here, this report called time course binning analysis is actually designed specifically for the visual world. It can be used for other paradigms, but it's designed for the visual world paradigm. And it's great for this type of stuff. So I want to come back to that in a second. You can find more details on reports in the video tutorial series, but for a visual world study, typically you're going to be creating interest area reports and time course reports, maybe trial reports too, but mostly these. So let's create an interest area report. And just as a quick reminder, before you create a report, you always want to make sure you've selected the interest period. That way you're only including the data from the relevant part of the trial. Okay, when you go to create the interest area report, you'll get a dialog that looks like this. And this dialog allows you to select what the columns in the output file will be. So each of these guys here is a possible column that can be included in the output file. Because we're creating an interest area report, we know that the rows will be interest area, or the cases will be interest areas, but the columns will be these guys, whatever we select. So these are the ones that are available, and these are the ones that have been selected to go into the report. So by default, you can see it includes the EDF file name and the trial number as trial index as the first 2 columns in the output file. But we probably want to include some other columns as well. So we probably want to include the interest area ID. So that was like 1 for target, 2 for phonological competitor, 3 for semantic, and 4 for unrelated. We probably want to include the label, which is those things that I just mentioned: target, semantic, or phonetic, semantic, etc. Then so that'll just give us, like, in the next 2 rows after the EDF file name and the trial number, the information about which interest area each row corresponds to. Then we probably want to do something like interest area dwell time, which would be the sum of the fixation durations for each interest area. And notice when you select a variable, you can see its definition down here. So dwell time is the sum of the duration of all fixations on the interest area. Okay, we could also do, for example, first fixation time. Let's include that. Note that this is the time of the first fixation relative to the start of the trial and not the interest period, but that's not a big deal because we can just include a couple other variables, and then if we wanted to get the time of this relative to the interest period, we can include IP start time, and we can include trial start time. And so then in the stats program, let me just bring up a notepad so you can see what, like, I'll make a new one here. Then if we wanted to get first fix time on interest area, that would be equal to the I'm mentioning this because a lot of times people want to do this. So you just do a little calculation in stats program. So it'd be like Okay, so basically we're just subtracting from that interest area first fixation time, the time from the elapsed time from the start of the trial until the start of the interest period. So that would just give us the time relative to the start of the interest period. Okay, in case you're interested in doing that. But I'll point that out, then let's include maybe just a couple more variables. So maybe like interest area run count. So this is basically how many times did you visit that interest area. A run in Data Viewer is a series of consecutive fixations on an interest area. So if you had 3 runs on an interest area, that means you looked at that interest area and you might have had separate fixations within that interest area, but then you left it and then came back to it, and then you left it and came back to it again. So you'd have 3 runs on it. So that's another, maybe we just include that variable. And you can see there are a lot of variables here, and I'll show you in a second where you can find definition, like a list of the definitions. But notice down here in blue, we've got all of our data source columns and variable nodes from our experiment builder project. So those become columns that can be included in any of the output reports that you create from Data Viewer, which is super important. So we can get our trial accuracy, like our reaction time, all these different, and then all our kind of condition information stuff here. So we actually had a variable called condition, and we had some other like columns that identified the images used. So I'm just going to select all those and maybe move those over. So now we got all our columns selected here, and you have some options down here. So I mentioned that by default, you're going to include the data from your currently selected interest period here in the report that you create. But if you check this, then you'll actually you'll include data from all the custom interest periods that you've created. In our case, we've created 2. And if you put them into one file, notice it added these extra columns that will tell you which interest period each segment of the output file kind of corresponds to. And if you do it into multiple files, it will still include those variables, but each interest period will be saved as a separate output data file. I'm not going to do that right now. I'm just going to do it for our currently selected interest period. You can also, if you wanted, you could create a separate report for each data file. Usually that's not such a great idea. Like kind of the power of Data Viewers, it allows you to like export the data for all participants, or one of the powers of Data Viewer. It allows you to create a report for all participants in one go. So I'm just going to leave that unchecked. There's also some other ways you can exclude trials that have a certain string in them, and you can do a little bit of formatting around your variables here. There's some more detailed settings in the preferences of the inspector. That's where you can find all the settings of your viewing session, but most of the time you can just use these defaults. Okay, anyways, let's create the report, and it actually reminds me I haven't saved this viewing session. So let's save the viewing session, and I'm going to save it in our little folder here, webinars, this one here, and I'll put it in the deployed folder along with the deployed project, and I'll just call it example viewing session. I'll call it example visual world viewing session. Okay. And when you save a viewing session, I just want to point out, it saves an entire folder actually. And in that folder, if you wanted to reopen the viewing session, this is you can double click this file. You can select this file from the file open dialog here. But basically this is the viewing session file, but it needs these other folders that are within it. So if you ever need to move the project around, don't just take this EBS file. You could actually use this file package command. I'll show you in a minute. But I just want to point out it creates a whole folder, and it creates an output folder that's relevant for what we're doing right now. And by default, when you go to save your actual report, it's going to choose that example view, like visual world viewing session output folder that I just showed you. It'll basically point to the viewing sessions output folder by default, which is kind of convenient. And I'll just call this interest area or IA report. Okay, and we just created the report, and so now let's go back to that folder, and there it is. And by default, they're always tab delimited text files. By default, it gives it an XLS extension in case you have Excel or some program that can open Excel files. So, but it really is just a tab delimited text file. And I'm going to double click on it since it's associated with Excel on my computer. But basically, it just gives you a little warning because it's not a true Excel file, it's just tab delimited text, but that's no big deal. Excel can figure it out. And so when I open it here, now we have our interest area report. And I'm just going to expand it so you can see the variable names at the top. So you can see for, I guess it left these IP variables in there, but basically each trial has 4 rows, one for each of our interest areas. And we got interest area ID, the label, the amount of time it's been looking at it, the first fixation time, all this different information that we included in the report. And so you could use this to kind of compare some basic measures across trials and participants. Okay, so that's kind of interest area report. There are a lot more options than what I just showed you. So let me go back here and mention, I said I would show you, if you go to help contents here, and then you go to data, and this is the data viewer, like help manual, user manual. If you go to data analysis and output, you can select on the different types of reports here. And if you scroll down, you can see all the possible variables and all the definitions of them. So this is kind of a nice quick reference for what these variables mean, and it can help you in selecting the variables you might want to include in your output file. So that can be useful. And one that I didn't mention actually is this IAFSA count, which is fixation sequence analysis. So this basically creates a different column for each interest area. We know that in the interest area report, each row is going to be a different interest area. So we would have a different column for each one as well. And the number for each cell would tell you basically how many transitions there were from the interest area corresponding to, I have to remind myself, the corresponding to the row to the interest area corresponding to the column. So how many fixations were there in the column interest area that were preceded by fixations in the row interest area? It's kind of like a transition matrix, which can be useful as well. But anyways, this is a nice little chart that shows you all the different variables that are possible for the interest area report. And I also mentioned if you're going to move around your viewing session, you don't want to just take this file. The best way to do that actually is to go File, Package, and this would actually zip that whole folder into one file with a DVZ extension. Then you can copy that file to another computer, email it to somebody, whatever you want to do. And then when they unpack it, they will have a complete and like full copy of the viewing session with all the interest area adjustments, anything that you've done in your viewing session, it will have that data. Let me also just mention, now's a good time to mention that anything that you do in Data Viewer has no effect on the raw original EDF files. So if you change interest area, like if you change, if you set interest periods, for example, or like change, there are ways to change fixation positions, things like that, or do filters on the fixations based on like durations and things like that, and those preferences, data filters. None of that changes the original data file. So you don't have to worry about anything you're doing in Data Viewer affecting your original data. If you want to start from scratch, you can always just start a new viewing session and re-import your data. Okay, so that's the basics of a interest area report. Now let's talk about this very cool report. Maybe cool is not the right word, but very useful report for visual world studies. It's called a time course binning analysis report. And this one is great. It uses basically the sample data, like so the ongoing eye position data, and it bins that data into, it kind of averages across bins of time to create what we call bins. And each row in the output file will correspond to a bin of time. And so if you set an interest period that is 1500 milliseconds long and you have a bin interval of 20 milliseconds, then that means you have 5 bins per 100 milliseconds, and we have 1500 milliseconds. You have 75 bins per trial, okay? So it's basically allowing you ultimately to create something that looks like this, okay? And well, in a stats program. So the idea is you would export a time course binning report, which would have the numerical data you need, import it into a stats program, and then create a graph that looks like this and do the number crunching, the statistics, inferential statistics that you need to do for certain bins in this graph. So let's talk about how this works. It's got a similar interface up here as the other reports, where you select what the columns are going to be. We know, because we're creating a time course binning report, that each row is going to be a bin of time, and you can specify the duration here. And we can select what the columns are going to be. So this is the EDF file name. We can have a trial label, trial index, and then we'll have a bin index included by default. So 1, 2, 3, 4 for each trial. And so each trial would start back over at 1. And this, and it would just, each row would have an incremental value. Then we have all these possible variables that can be included. But it's kind of special in that it has all these variables that have stars on them in their name. And those variables will create a separate column for each interest area, okay? And by default, it will use interest areas with IDs 1, 2, 3, and 4 when it creates the output report. So by default, it's by default set up to use 4 interest areas. And that's perfect for this because we have 4 images with IDs 1, 2, 3, and 4. But if you had more, like let's say you had 8, then you could either type just an 8 there, and then it would actually include 1 through 8, or you could do 1, 2, 3, 4, 5, 6, 7, 8, or you could type in the labels separated by commas. So we have a label target, phonological, competitor, etc. You can do it that way too. And if you want more information on that, you can go to the same section of the data viewer user manual, go to time course binning analysis. If you scroll down here to the very bottom, there's a little section on interest areas used for calculation that describes how you can specify the ones. But by default, it's going to use the interest areas with IDs 1 through 4. So I'm going to include, I'm just going to leave it blank, so because we don't need to specify anything differently. And sorry, I didn't really exactly explain what this is. So I mentioned it's going to create, based on what your interest areas you selected, a different column for each of those interest areas. In the case of like average IA star sample count, we will have actually 5 columns. Average IA1 sample count, average IA2 sample count, 3, 4, and then average IA0 sample count. And the 0 one will the column with a 0 in the name will be for data that is not on any interest area. So if they're just looking between the interest areas, like here or off the screen, or what, depending on your settings here, then it's going to have data for that. And this sample count one means within that bin, how many samples were there in the interest area. And the sample count percentage will mean within that bin, what percentage of the samples were in that interest area. And we'll talk about how the proportion or percentage is calculated in a second. The average means that it's using data it's averaging the data from the 2 eyes, from the left and the right eye. So if you scroll down here, you'll see you have, since this is based on sample data, we can potentially have data from the left eye and the right eye. And so those can be included as separate columns. If average is selected as, if these average variables are selected to be included, then usually for a visual world study, the best thing to do is turn on what's called cyclopean mode. So let's imagine we recorded monocular data. So we either recorded from the right eye or from the left eye. If cyclopean mode is turned off, then when you get these average variables, you'll have missing data because there's no average of one eye and no other eye, right? You can only average if you have different values. But if cyclopean mode is on, then if you have monocular data, it will take like the right eye and include that for the average variables, and the left eye and include that for the average variable. Like if you track the left eye, it will include data from the left eye and the average variable. If you track the right eye, include data from the right eye and the average variables. So that way you don't have to worry about doing any extra selection in a stats program to say, okay, for this participant, we tracked right eye, and so I need to select the data from the right columns. In other words, let me get back here. So as I mentioned, we have these left eye variables and these right eye variables, and we don't need to worry about it if we do average and we track monocularly, because this will already kind of do that combination for us. And actually, if you have that turned off in this newest version of Data Viewer, then when you run it, it'll say, are you sure you want to do that? You have monocular data, then it will be missing. But since this is on, we're going to have data from both of them. Okay. Let's also include average pupil size for the bin, something like that. These are the main ones. You can also get some blink data and excluded data, something like that. I'm just including some for just an example purpose. And then we can, if we wanted to remind ourselves what the numbers in this star name correspond to, this IA star ID will give us information about that. Let me see if there are any others we should include. Okay. Let's include down here our data source columns and variable nodes from our experiment builder project. You can include lots of other information here. And again, you can check the manual for that. Now let's talk about these options down here. So we talked about these up here, or we talked about this one and this one. This maximum number of bins, let's imagine in a given trial, maybe the participant just took a really long time to respond, maybe several seconds, and you just want to cut it off. You don't want to even look at data past 2,000 bins for a given trial. So you could specify a maximum number of bins for each trial. Then down here, you know how I talked about these variables like this one that are going to compute within a given bin what proportion of the samples were on each of the different interest areas. This determines how we're gonna what's the denominator for that calculation, basically. So if you have across all samples, it doesn't matter like where the samples were within the bin. It's just going to be divided by the bin interval. Okay. If you have across all on-screen samples, then if the participant was looking like off the screen and you have data that's outside the screen bounds or missing data, like for blink, for example, then let's say you have within our 20 millisecond bin, we have 6 milliseconds of blink time where there's no data for those 6 milliseconds. So in that case, we would, for that bin, be dividing by 14 rather than by 20, okay? And if you do across all samples assigned to predefined interest areas only, then you're basically gonna exclude any samples that were on screen but outside of the interest areas in your proportion calculation. So this is kind of gives you some nice ways to filter. For now, I'll just use across all samples. Then in terms of the data selection, sometimes people want to exclude samples that were part of a saccade, maybe for reasons of like saccadic suppression or something like that. And so basically you can check this box and exclude samples that were part of a saccade. I'm going to just do the default and include all samples, fixation, and saccade. Okay. And you can also, if a bin just had any data at all within it that was not part of a non-fixation, like either part of a saccade or blink, then it's going to just basically exclude the whole bin. So that's kind of even more strict. Okay, down here you have the normal exclude trial string, which is very rarely used, but it's a way you can exclude certain types of trials. And then here, do you see create a separate report collapsing data across trials in the same group? Okay, let me go back here for a second. So we talked about how you can regroup the data here. So I right now regrouped it by identifier. Then that means we're going to have a separate output file that's going to do the averaging for each bin for each group of trials. Let me show you a simpler one. Let's regroup it by like condition. Okay. So now I had condition in this example A, B, C, and D, and then the training. Okay. And so in this case, these are like the practice trials. So let's imagine we have an independent variable with 4 possible values, and we want to just go ahead and help like get data viewer to do some of the averaging for us across the different conditions. If you have that property checked here, then we're going to get a separate report that's going to do that collapsing for us. And then again, we have our ability to create a separate report for each custom interest period. So right now I'm not going to use that. So we're basically only going to have 500 milliseconds before the critical word to 1000 milliseconds after the critical word. We're going to have these columns in it. Let's do it. Let's save our viewing session first. Okay, and then I'll call this time course report. Okay, and it creates a report. And see, it actually created 2 reports. One, which is going to be collapsed across all the trials in the group, and one for all data separately for each trial. So let's open that one first. And we can see that we have all our different bins for a given trial here. And it starts at 0. So I mentioned we're going to have 75 bins, and we go 0 to 74. So that's 75 bins. Okay, that's all for trial 1. And then you can see we have average IA1 sample count, average IA2 sample count, and you can see over the course of the trial they're looking probably at they're probably looking at the center of the screen from the cross or something maybe on that trial. Then they move over to interest area 2, move over to interest area 3. So this is target, phonological, semantic, and unrelated, and this is off all interest areas. And then we have our percent columns here and that kind of information. And so you can see they're actually proportions, not percents, but you can see that there. And then you can see the pupil size for the bin. And then as we go to the right, we can see our data source columns and variable nodes from our experiment builder project. So that's the time course report, super useful. Let's take a quick look at the collapsed one. And if we take a look at this one, you can see this is all for group A. We have through 74. Okay, so it's averaged across all the trials that are in that group across participants. And if we have multiple trials in a given viewing session for a given participant, then it's like collapsing across all the trials within the participant that fall into that condition. So it's basically all the A trials in the whole viewing session. And then we have all the B trials for the whole viewing session. So it's already averaged across condition for us. So this can save you a little bit of time. And then it's got the same data that we have up here. Okay, and we got our interest area IDs here. I didn't point that in the report, but that can help you remind you which of each of these columns refers to. So then you have this data that's ready to go in a stats program, and from there you can do your statistics and create those graphs. And I think that's everything that we probably should cover today. Hopefully this helps. If you have any questions about how to do visual world analysis, how to use data viewer in general, or really anything else related to any of our software or hardware, don't hesitate to get in touch. You can send us an email to support@sr-research.com, or you can go to our support forums that I showed you previously, and you can post on there, you call us, whatever you want to do. Don't hesitate to get in touch. We'll be happy to help you with any of your questions. good luck out there in your visual world, data analysis, and anything else that you're doing. And thanks for joining today. And that's it.